Okay, so I'm going to go ahead and turn this over to Eric, and he's going to take you through the Datasets command line tools. So take it away, Eric.
All right, thank you, Peter. Thanks everybody for coming today. Welcome to the webinar on using Datasets command line tools to download and use genome metadata. So here's an outline of the webinar today. First, I'm going to introduce the Datasets project, then our command line tools, and then I'm going to go over 2 examples of using our command line tools. So first, I'm going to show you one way to get high quality butterfly genomes. And then I'm going to show you how to find SARS-CoV-2 lambda variant genomes. So the butterfly genome example will hopefully give you some general ideas about how you can get genome metadata and sequence data for really any organism, while the SARS-CoV-2 example is a little bit more specifically focused on SARS-CoV-2 data. So Datasets is a relatively new effort at NCBI that's focused on making sequence data easy to find and download. We have web pages, including our homepage shown here, command line tools that I'm going to talk about today, and a documented public REST API. A big emphasis of the project is providing easier access to genome data, including genome sequence, genome annotation, and transcript and protein sequences. So today's presentation is just focused on accessing genome data using our command line tools. So as I said, we're looking at 2 examples of using our command line tools. In both examples, we'll use the Datasets tool. This is for downloading biological sequence data. And in the second example, we'll also talk about data format, which is used to generate human-readable metadata tables. We don't have time today to go over installing the tools, but you can download them from the Datasets website, and we've recently made them available through Conda. We're also going to use 2 other third-party tools, mostly JQ, and we'll also touch a little bit on SAM tools. JQ is a great tool for working with JSON formatted data. The Datasets command line tool delivers metadata in 2 flavors of JSON and JSON lines. So just a quick note about versions. Today we're talking about version 12 of the Datasets command line tools. These are expected to remain stable for the foreseeable future, and these are based on version 1 of our API. And at the moment, we're starting development on API version 2, which will power future releases of the command line tools. And we're planning a lot of new features and some breaking changes. This is a great time to give us feedback on where we'll be going next as we start planning version 2 of the API. So for the first example, we're going to download metadata describing butterfly genomes, then use information in this metadata to help us identify and download a subset of high-quality genomes. We're going to use a built-in filter to get butterfly genomes at the chromosome assembly level. Then we're going to further restrict this set of chromosome-level butterfly genome assemblies to those above a certain contig N50 value. Along the way, we'll show you how to get genome metadata, how to convert genome metadata into human-readable formats, how you can filter the metadata, and finally, how to download a set of genome assemblies. So we're going to start by downloading genome metadata for all butterflies. So we're going to go ahead and switch to the terminal window now. Okay, so one way to get genome metadata is by using the Datasets summary command. So I'm going to go through each part of the command and describe what it means. So Datasets summary means that we are going to get metadata. Genome specifies the type of metadata, and taxon tells you what kind of query you're going to use to get this genome metadata. So this can be a taxonomic name or an NCBI taxonomy ID. In In this case, we're just going to use the name butterflies. The assembly source parameter allows you to specify whether you want data for either GenBank or RefSeq assemblies. Today, we're just going to look at GenBank assemblies to keep things simple. And we're going to run this command and save the output to a file, butterfly summary.json. So let's start with a simple question about this metadata. How many genomes are there? So here's one way to get the genome count. We're going to use JQ to pretty print the data to make it easier to read. And then we're going to pipe this to tail to show the last 10 lines of this pretty printed data. Because the genome count is always shown at the end of the genome metadata that you get using the summary command, this command will let you see the number of butterfly genomes described by the metadata. So you can see here, it just shows total count 562. So that means there's 562 genomes. Another way to get the genome count is to specify the name of this total count field, and using the power of JQ, just pull the value out directly. So here we just run JQ and specify the field name, total count, and the file name. And then you'll just get that genome count back directly. So since we're looking for high quality butterfly genomes, we can use the assembly level filter to only get chromosome level butterfly genome assemblies. So we're going to run this summary command again, but this time we'll use two filters. One filter to get GenBank assemblies, and one filter to get chromosome level assemblies. Okay, so we've saved that metadata to a file, butterfly chrome level.json, and now we're going to use JQ, specifying the field name of total count to see how many genomes, how many butterfly genomes are at the chromosome assembly level. So you can see that number is a lot smaller than what we had before. So we've reduced the set of genomes from almost 600 genomes to less than 50. So it's great when you need to filter by some attribute where we already have a built-in filter, like assembly level or assembly source. We also have filters to only get annotated genomes or get genomes released before or after a certain date. But sometimes you might need to filter by other attributes of a genome record where we don't have built-in filters available. So I'll show you how to do that using JQ. People often use contig N50 value as a measure of genome quality. So to further narrow down our set of butterfly genomes, we're going to take the list of chromosome level butterfly genome assemblies we already have, and then only keep the genomes where the contig N50 value is over 1.5 million. So I'm going to show you how to make a simple table of assembly accessions and contig N50 values using JQ. So basically what this command is saying is take this metadata describing the chromosome level butterfly assemblies, send it to JQ, and then for each assembly, give me back the assembly accession and the contig N50 value, and then generate a tab delimited table in TSV format. And then I'm going to pipe that to head to just show the first 5 rows of that table. So now you have this two-column table. The first column has assembly accessions. The second column shows you the contig N50 values. So next, we're going to get the genome accessions for those genomes with a contig N50 value over 1.5 million. So again, we're sending the metadata for all chromosome level butterfly genome assemblies to JQ. Then this JQ command is saying, for all assemblies in the set, just give me back the assemblies with a contig N50 value over 1.5 million. And then for each of those assemblies, just give me the assembly accession. So this is the whole list of butterfly genome assemblies that are both chromosome level assemblies with contig and 50 values over 1.5 million. And in case you're curious where these genomes came from, these are all from the Darwin Tree of Life project, which is a project to try and sequence 70,000 eukaryotes from Britain and Ireland. So next, we're going to save this list to a file. So I'm just going to run that same command again and save it to a file called accession list. Now that we have this list of assembly accessions, we can download the genome sequences for these genomes. So this takes a few minutes, so I've already downloaded it in advance, but I'm going to show you the command that I used. So this is what the download command looks like. I'm going to go through each part of the command and explain what it means. This says download genome data given one or more accessions. In this case, we're using a text file with the list of accessions we generated previously to specify which genomes we want. So then why do you need all these exclude flags? So Datasets delivers our sequence and annotation data in a zip archive that we call a data package. By default, these packages will include a bunch of different types of sequence data, annotation data, and metadata. So for example, for an annotated eukaryote genome, we would include the genome sequence, transcript, and protein sequences, annotation in GFF3 format, and metadata in one or more files we call data reports. So if you only want to get genome sequence, you can add various exclude flags to, for example, exclude the protein sequence, exclude the annotation file, and exclude the RNA or transcript sequences. So let's take a quick look at what's in this downloaded package of genome sequence. I'm going to use the tree command to show you the directory structure and some of the files that are included in the download package. So as I said, I've already downloaded this zip archive and extracted it on my computer. We're going to run tree on this directory that contains the contents of that zip archive, and then pipe that to less so we can scroll through that output. So you'll notice that data for each genome assembly is in a directory that's named with that assembly accession. So in this case, we asked for just genome sequence. So you'll see FASTA files in each of these directories representing each assembled chromosome. Sequences from unplaced and unlocalized sequences are in separate files. And in each directory representing a genome assembly, there's a sequence report file. That's basically a list of all the sequences that comprise the assembly. And then if you scroll all the way down to the bottom, you'll see the assembly data report, which describes all the genome assemblies that are included in this package. So that's the end of the first example. Okay, so next we're going to talk about SARS-CoV-2 genomes. So this example is focused on SARS-CoV-2 genome data. We'll show you how to download genome data and view and filter the associated metadata. Specifically, we'll show you how to pull out SARS-CoV-2 genome metadata and sequence data for one SARS-CoV-2 lineage, the Lambda variant of interest. So first, I'll show you how to download data for all SARS-CoV-2 genomes. So this is the command you'd use to download all SARS-CoV-2 genomes. We're now up to over 1.5 million genomes. So this download can take a little while depending on your internet connection. For the purpose of today's demo, we're going to use a smaller package of SARS-CoV-2 genomes that were released during a 3-day period last week, which contains just about 65,000 genomes. So I've already downloaded the zip file and extracted the zip archive. So we're going to use tree again to take a look at what's inside what I've downloaded. So you'll see there's just a few files in the data directory compared to that genome data package that we downloaded in the last example. I just want you to pay attention to two files here. There's the data report that includes metadata and annotation data for each viral genome, and genomic.fna includes all 65,000 genome sequences. So now I'm going to introduce the other command line tool that we've developed. It's called data format. This is specifically for generating tables based on the metadata in this data report file here. So we'll go through each part of this command and what it means. So data format is the name of the tool. TSV means we want the output to be a tabbed limited table. And virus genome specifies the type of data report we'll be using as input. We'll use the input file parameter to specify the path to the data report itself. And then we'll use fields to specify all the columns that we want to include in our table. So here we're including isolate lineage, which is an identifier for the genome record. Accession is the NCBI nucleotide accession. Geolocation is the geographic location where the virus was isolated. Isolate collection date is when it was found. And virus pangolin is the pangolineage name that we've calculated for that genome. And then we're going to use head just to show the first 5 rows of that table. So you can see there's 4 genomes here. It's a little bit hard to read. The last column is kind of going over to the next row. But if you've looked at these pango lineages before, you might recognize this B16172 as the so-called Delta variant. Next, we're going to pull out metadata for all SARS-CoV-2 genomes representing just the Lambda variant of interest. So one thing you should know about our data reports is that each line of the data report represents a single record, or in this case, a single SARS-CoV-2 genome. So this allows you to use grep with an identifier other string to pull out all the metadata for a particular genome containing that identifier. So in this case, we want to get all the metadata for the Lambda variant of interest genomes. We don't have the WHO term Lambda in our data reports, but we do have the pango lineage names. And for Lambda, that's C37. So we'll grep for C37 to pull out the metadata for those Lambda variant genomes. And then we'll save that to a file, lambda.jsonl. So now let's use, it's taking a little bit longer than expected. There we go. All right. Now we're going to use data format and take a look at the metadata we've saved. So we're going to basically run the same data format command that we just ran, but now we're going to use this new file that we've generated as the input file. So let's see what that looks like. All right, you can see that if you look in this last column, you can see now we only have the Lambda variant genomes, or the C37 genomes. Next, I'm going to show you how we can use data format and SAM tools to pull out the genome sequences for these Lambda variant genomes. So first, we'll use data format to get the accessions only from our metadata file. Then we'll use that list of accessions to specify which genome sequences to pull out of our genome sequence file. So first, we'll use data format again. This time, we'll specify just a single field accession to get a single column table of accessions only. And we're also going to use the ELIDE header flag to remove the column name, which could cause problems with SAM tools. So I'll just show you what the first 5 accessions looks like. Okay, now we're going to go ahead and save that list as a file, lambda accessions. Now we're going to use SAM tools to extract the lambda sequences from the original genomic FASTA file that we downloaded as part of the package. So here is the SAM tools command we're using. We're going to specify the file with the accession list. This is the original genomic sequence file. And then we're going to save the output as lambda genomes.fna. All right, and then let's see what you can do with that file. Okay, we're going to just drag and drop that FASTA sequence file into the Nextclade website. You give it a few seconds. This gives kind of a nice way to visualize these genomes. And it's nice to see that they've also identified these as lambda variant genomes. And this kind of gives you a nice visualization of other mutations that are present in the genomes and some ideas about genome quality. And that concludes the second example. So I think we can take some questions.
Hi, Eric. Thanks. That was great. There's a few questions. One, there was a little bit of discussion about installing this on a cluster, and I wanted Brad to comment on that because he had a couple of thoughts about that. Brad, are you there? Brad Holmes?
I am, yeah, happy to speak to that. So I'd be honest, I'm not, we have a different kind of cluster here, and in our case, we'd be responsible for either every time we submit a job, ensuring that the app gets installed from Conda or through some direct download link, the quick starts link there that's also in the answer. So that's one option, or you can talk to the sysadmins and say, look, we want this sort of globally installed, just like you install any other package, and they could follow the similar steps to ensure it's available on every machine. So I think the answers kind of sort of vary on what the administrators want to do with the cluster.
Okay, and you had sort of had some comments about rate too.
Oh, thanks. Yeah, sorry. So yeah, that's the other thing is these tools are rate limited. It's possible to get an API key. We're working on setting up API keys through my NCBI, which would allow a slightly higher rate, but we do not ever intend to support like 400 concurrent connections from the same API key, all downloading a package. So it would be bad practice to say, all right, well, I want to start 400 jobs and they all go download, a million sequences, the same million sequences, and they sort of in parallel operate on that package. It'd be better practice to have a single task to first go get that package of data and then send that data out to the 400 jobs or in however you share data on your cluster, and they can then operate on the same package. So if we do see sort of, service is going to degrade if there's that many concurrent connections coming in.
Okay, thanks, Brad. Another question that was here is about downloading by accession.version, and Brad chatted to me that he didn't think that was possible. So, Eric, do you have any comments on that?
Nice.
In other words, can you just specify the access? I think this is what the person wanted to know. Like, can you just give it an accession.version and use datasets to download something that way?
You can only for with assembly accessions, genome assembly accessions, but you can't do that yet for SARS-CoV-2 genomes.
So
you can also download gene data by refseq accession.versions.
Good point.
But yeah, the question came in while we were looking at viruses, and so in terms of the virus genomic refseq accession, there's not a way to access that data. If that's useful, we'd like to know.
Okay, and there was another well, here's a couple more that have come in, but one of them was had to do with how quickly sequences that are submitted, I think is what the question is really about, how quickly they're turned around. So when we're talking about SARS-CoV-2, do we have any, and this may be a question for another group at NCBI, and if it is, I'll get the answer from them too, but how quickly does a sequence get from being submitted to being available in data sets for the SARS-CoV-2 sequences?
Hi, I can answer that. So when the sequences get submitted to GenBank, they then get picked up by NCBI virus, if you're aware of that resource, and we pull them from NCBI virus. So Brad maybe could be able to comment on exactly how long that takes, but we do update our virus data set every day. So we should pick up anything that's new in NCBI virus, and I believe that also gets updated every day, or a couple of times a day. I don't know, Brad, if you have any more information on that.
I don't. Yeah, it's so many factors involved. How quickly the labs turn over the data, how quickly it goes through our submission system, how many other submissions we receive the same day. Well, we're working to get it out as fast as possible. 24 hours is certainly possible, but there are no guarantees on that.
And then somebody asked a question that is probably worth addressing. You know, you mentioned that there were 1.5 million genomes for SARS-CoV-2. And the question is, are they all unique or are they duplicate sequences isolated from different individuals? Oh, and Brad sort of answered that, but they're basically different samples.
They're all unique submissions. The sequence could be identical.
Right.
But each submission is unique, yeah.
Okay, and there was another question that, just a general one about how to find out about webinars and workshops like this. And Brett, Eric, did you have that slide at the end that had the sort of links to the social media and things like that? I don't know if we kept that in your slide set or not.
Yes, here we go. This one?
Yeah, so yeah, just to point out to you that, you know, we have social media presence and, you know, we announce all these things on social media. So on Twitter, on Facebook, and on LinkedIn. We also have a blog, NCBI Insights. And so if you go to that blog and then you have a WordPress account, you can subscribe to that blog as well. There's also an old-fashioned email list that somebody asked about, and that's the NCBI announce email list. So if that's something you want, I can put the URL and the chat to everybody, and that's how to get to that if you want to subscribe to that email list. Okay, and I think we covered all the questions that we have in the chat right now. And if there's any that we didn't get to answer, I'll make sure that they're in the document. So thanks everybody for coming. And thanks Brad, Nuala, and Eric for being here.
Thank you.
Okay, thanks a lot.